ब्रह्मनाथ शांति 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 स्वस्थ।
Hello students, in the previous class we discussed about the composition of the blood portion, that is your blood platelet. We discussed that in the case of the blood platelet, it have the diameter around 2 to 4 micron. Its structure is somehow we can say rounded, colorless, non-nucleated. Okay, and also we discussed that the range which varies in between the 2.5 lakhs to 3 lakhs per cubic mm of the blood students. Okay, next, also the shape of the blood platelets are somehow we can say biconvex in shape. Okay, next we discussed that the lifespan is about 10 to 12 days. Next, if the count of the blood platelet increases, then that condition is known as your thrombocytosis, and if the blood platelet count decreases, then that condition is known as your thrombocytopenia. Okay, and also we discussed that it is being developed in the giant cells of the in the bone marrow. Okay, next also we discussed the ABO blood group which is one of the important short notes. In the ABO blood group we discussed that it was given by Carl Landsteiner. Okay, in that also we discussed that the RBC have a particular antigen, means in a particular blood group it have a particular antigen, and against to that antigen or we can say opposite to that antigen, antibody is being present in the plasma. We discussed all the points AB and the O blood group, which antigen is present in which blood group, and opposite to that which antibody is present. We have discussed all the points. Next we discussed that the which blood group can receive which blood from which blood group, or we can say the particular blood group, the person having a particular blood group can receive the blood from which blood group, or he can give the blood to which blood group. By this we came to a conclusion that the AB or the blood group or the persons having the blood group AB can receive the blood from all the blood groups. That is why they are called the universal recipient students. And as the blood group O or the persons having the blood group O can give blood or donate blood to every blood group, that is why they are called the universal donor. Okay, AB blood group can receive blood from all the blood groups, that is why they are called the universal recipient. A blood group can give the blood to all the blood groups, that is why they are called the universal donor. Okay, AB universal recipient, O universal donor. So this much we discussed. Then we discussed another blood group, that is your RH blood group or the rhesus antigen presence in the blood group. If the rhesus factor is present, then that person is known as RH positive. If absent, RH negative. And also we discussed the compatibility of the marriage in between the boy and the girl. Next we discussed that this rhesus antigen was first discovered by in the case of the rhesus monkey, and it was also been given by the Landsteiner. Also we discussed in the two cases how the problems may occur in the rhesus means we can say in the presence of the rhesus antigen factor. There are two problems, condition in the two problems can arise during the blood transfusion case and also during the pregnancy case. We have discussed in detail about the pregnancy case, also about the blood transfusion case. For details, you can go back to that video and also watch the entire video regarding the blood transfusion case and the pregnant case. Okay. Students, today we will discuss that as we know students, the blood platelet also might remember that it is responsible for the blood coagulating or the blood clotting. I have said you. So, what do you mean by blood clotting, students? See, whenever any injury has been cut in our body, what happens? Blood regularly flows. But does it flow continuously? Does it flow regularly for prolonged period, for 1 hour, for 2 hours? No. After 2 to 3 minutes, or in some person after 5 minutes, the blood flow will stop. And that stopping, or we can say that stoppage of the flowing of the blood, by which process it is being stopped, or by which process the blood flow is being stopped, that is known as the blood clotting. Okay students, once more I am telling you, the blood, whenever any injury has been done, the blood generally flows. Whenever that blood flows up to 2 to 3 minutes or the 5 minutes, after that you will see that the blood will not flow and the clot will be formed. The process by which this stoppage of the blood forms, or we can say occurs, or the clotting of the blood occurs, that is known as your blood clotting or the blood coagulation. Today we will discuss and focus on that portion, how the blood clotting, how the blood coagulation takes place. It generally takes place, students, in regularly 3 steps, and there are total of 13 factors that are being responsible or required for the blood clotting. Okay students, and the recent blood clotting theory, which is known as we can say the enzyme cascade theory, this was given by the McFerrin, according to which 13 factors are being required for the blood clotting. Okay, starting from the factor 1 to the factor 13. Okay, and these all are, students, written always in the Roman letter. Roman means in this language, not 1, 2, not like this. Roman language means in this manner. Okay. You should always remember, students, this one. If you can remember, then it is sufficient. More often, if you want to remember, then you can remember some important, such as the thromboplastin, such as the fibrinogen, such as the Christmas factor. Just 3 to 4 important common name you can remember, but you can only remember only the name of the factors by their number. Okay, so for as of now, I will tell you, factor 1 is the fibrinogen, factor 2 is your prothrombin, factor 3 thromboplastin, factor 4 calcium, factor 5 proaxlerin, factor 6 is your dilated, factor 7 it is your proconvertin, factor 7 is your proconvertin, factor 8 it one is your anti-hemophilic globulin or the AHG. Okay, what is AHB? I will tell you, don't worry. Factor 9 Christmas factor, factor 10 Stuart power factor, factor 11 plasma thromboplastin antecedent, or it is also called the PTA, factor 12 Hedgeman factor, factor 13 fibrin stabilizing factor. So these are the 13 factors that are required for the blood clotting. HA, HB, HC, what it is, I will tell you, don't worry about it. Okay. So what we have read now, see, I am telling you once more time. When any injury is being caused by the blood vessels, bleeding occurs for some time, and after that the clotting occurs by a process, and that process is known as blood clotting or the blood coagulation. As I said you, suppose here a cut arises. Obviously, blood vessels are all over our body. So if that portion is being cut off, obviously the blood vessels will also cut off. As the blood vessel will cut off, bleeding occurs. But for some time, after that the blood clot occurs, or we can say stoppage of the blood flow occurs. By which process that occurs? That is nothing but the blood clotting or the blood coagulation. Blood clotting or the blood coagulation. Students, it generally involves 3 steps. Okay, see how it is being formed. See, if a cut happens, what happens? Tissue will damage, or we can say the tissue will injure. Yes, that injured tissue will release the factor number 3, that is called as the thromboplastin. And also at the site of the injury, blood platelet will disintegrate, and also they will release the platelet factor 3, that is called a thromboplastin. That is your thromboplastin. You can see here factor number 3, thromboplastin. The thromboplastin, which is being released from the injured tissue and the blood platelet level, they, in the presence of the calcium and the certain protein, they helps in synthesis of the prothrombinase. That prothrombinase, students, in the presence of the calcium, okay, it will forms the prothrombin, which is the factor number 2. See, factor number 2, that is your prothrombin, and it is basically in the inactive form. Okay students, then further what happens, that prothrombin is also in the presence of calcium does another function. What it does students? See, it inactivates the heparin. Now the question arises, what is heparin students? Heparin is a substance which is present inside our blood, in the blood vessels, which prevents the blood clotting. Otherwise students, in our body also blood flows. Why the blood clot is being not formed inside our body? Why the blood clot is always formed when the blood oozes out of our body? The reason is the presence of heparin. This is the substance which prevents the clotting of the blood inside the blood vessel inside our body. Okay students, so this is one function, that the prothrombinase in the presence of the calcium, it inactivates the heparin, by which the clotting can be now happened. And also in the presence of calcium, it will form the inactive prothrombin, that is the factor number 2, okay, which will further be converted into active form, that is called the thrombin. Okay, this is one, we can say step 2. Step 1 is from up to the release of the factor number 3, that is the thromboplastin factor, step 1. The step number 2 is from the thromboplastin in the presence of calcium protein, it will form the prothrombinase. Okay, and this is being formed in the, we can say, the prothrombinase in the presence of the calcium. Okay, it will form the prothrombin 2. Okay, and this is being done in the liver in the presence of the vitamin K. Further, the prothrombin 2, which is inactivate form, it will convert it into active form, that is known as your thrombin, and this much is your step 2. Finally, what happens students? The thrombins, they catalyzes or depolarize. Means what? Depolarize means it will separate the substances, the fibrinogen into fibrin. Fibrinogen means you can see the factor number 1. It will, what it will do, the fibrinogen which is present, it will depolarize it. Means the fibrinogen is in bundle like manner, it will depolarize it into fibrin manner, means thread like substances, those are called the fibrin. Depolymerase means what? To get together. Depolarize means what? To separate them. So that is why the thrombin, which catalyzes or depolarize by the help of the depolarize, not depolymerize, by the help of the depolymerize, it converts the fibrinogen into thread like substances called the fibrin. Finally, when the fibrin comes in contact with the blood cells, the blood clot happens, and this much stage from the thrombin up to the clot is forming, this is known as your step 3. Okay students, so in the blood clotting, 3 important steps are being responded. Okay, or 3 important steps are present. First, the release of the thromboplastin. Okay, next the synthesis of the prothrombinase and the conversion of the inactivated prothrombin into thrombin. This is one is step 2. Step 3, from the thrombin, it catalyzes or depolymerase occur, by which the fibrinogen is converted into fibrin, and the fibrin, when mixed with the blood cells or come in contact with the blood cells, the fibrin plus the blood cells, finally the blood clot happens, and this final stage is known as your step 3. So you can see step 1, step 2, step 3. That is why I am written, it generally involves, or the blood coagulation or the blood clot generally involves the 3 steps. Okay, first we discuss the definition, then we discuss so many that factors that are present. Don't worry, HA, HP, HC, I will tell you. Okay, and also we discuss the 3 steps that are being involved in the, we can say, blood clotting. Okay students, please note down. Students, now come to some important terms that are related to the blood clotting. Students, there are some particular substances which are being used in the blood bank to prevent the blood clotting. What happens students? See, I am telling you. In the blood bank, we know that there are separately the blood group wise, the separately the blood are being stored. As we have discussed, if the blood comes out of the body, obviously the blood clot will happen. Okay, and then that blood or that clotting blood cannot be used obviously, so we should use the clotting free blood, or we can say the blood in which no clotting should be done, no clotting should be occurred. So in that case students, what happens? While storing the blood, obviously as the blood came out of the body, clot might would happen. But what it should be done so that in the blood bank the blood should be present without any form of the clotting. So students, in that case some substances are being used to prevent the blood clotting, such as the CPD, citrate phosphate dextrose, then the ACD, acid citrate dextrose, EDTA, ethylene diamino tetraacetic acid. These are the basic substances, students, which are used in the blood bank to prevent the blood clotting. These are used in the blood bank to prevent the blood samples from the blood clotting. Students, if we can say blood clotting happens inside the blood vessels, then what we will tell? Then if the blood clotting happens inside the blood vessels, then that is known as, the condition is known as the thrombus, and by which process it is being formed, that is known as we can say thrombosis. Okay students, next point. The blood clotting is prevented in the test tube. Suppose we are in for testing, we are taking in the test tube the sample of blood. So what we can do so that the blood will not clot inside the test tube? Yes, by adding some sort of oxalate or the citrate. Okay students, now come to the hemophilia. Students, hemophilia means it is a bleeding disorder in which the blood clot never happens, or we can say for a prolonged time the blood is being flown out of the body. What we generally know that in the case of the blood clotting, after a cut or in the tissue level or in the in the body, if a cut happens, blood flows. After 2 to 3 minutes or 5 minutes the blood clot will happens. But if that blood will flow for some several hours, then that disease is known as the hemophilia. It is of 3 types, students: hemophilia A, hemophilia B, hemophilia C. Now come to that point, HA, HB, HC. Yes, HA means hemophilia A, HB means hemophilia B, HC means hemophilia C. As we have discussed, in various 13 factors are there which are required for the blood clotting. If factor 8 is absent, it will cause the hemophilia A. If factor 9 is absent, then it will cause hemophilia B. If factor 11 is absent, then it will cause the hemophilia C. That is why I have written in the factor 8, HA means hemophilia A, factor 9, HB means hemophilia B, and factor 11 means HC means hemophilia C. Means what? If factor 8 is being absent, then it will cause hemophilia A. If factor 9 will be absent, or we can say deficiency of factor 9 will cause the hemophilia B. Deficiency of factor 11 will cause hemophilia type C. So students, hemophilia means it is a bleeding disorder in which the blood clot never happen, or the blood clot which does not happen, or we can say the blood flows from out of the body during some injury for so many prolonged time, or we can say the blood flows continuously for several hours, by which most of the measure of the blood loss can be occur from the body, and lastly it can cause a lethal or fatal, means what, the death of that person can also cause. Obviously students, if the blood will not clot and more amount of blood will flow, then what it will happen? Then obviously heavy blood loss will occur, by which the person can also die. Okay students, so these are the some terms you should remember, such as the CPD, ACD, which is used in the blood bank to prevent the blood samples from clotting. Thrombus, clot forms inside the blood vessels. That process known as thrombosis. And also blood clotting is prevented in the test tube by adding little amount of oxalate and citrate. And also finally we discuss the hemophilia, which is of 3 types: hemophilia A, hemophilia B, hemophilia C. Hemophilia A caused due to deficiency of factor 8. Hemophilia B caused due to deficiency of factor 9. Hemophilia C caused due to the deficiency of factor 11. Factor 11. Okay students, please note down. Students, now we will discuss various pathway that has been related to the blood clotting. Students, as we discussed how the blood clotting occurs, these are being occurred by the following 3 steps. But the step 1 what we have discussed, in the tissue level the injury occurs, okay, that releases the thromboplastin, okay, that thromboplastin the factor number 3, okay, and also in the blood platelet level also the blood platelet disintegrate to form the or releases the factor number 3, that is also known as your, we can say, thromboplastin. So main question arises, students, this thromboplastin further it will help in the blood clotting, but how this thromboplastin is being formed? These are being formed by the help of the various pathway. If we are discussing the formation of the thromboplastin in the blood vessel, then we are discussing the intrinsic pathway. If we are discussing the formation or the releasing of the thromboplastin in the tissue level, then we will discuss the extrinsic pathway. Clear, students? Now let's first discover in the blood vessel how the thromboplastin are being formed, or we can say the intrinsic pathway. Okay, so what happens? You listen carefully. First of all, injury occurs. Injury occurs means blood vessels is also cut off, by which the blood platelet disintegrate. We know the blood vessel is being internally lined by the endothelial cells. What happens? First, at the time of the cut or the injury of the blood vessels, the surface alteration of the endothelial cells takes place. Means we can say the change in the endothelial cells takes place, by which what happens? Factor number 12 gets attached to the site of the injury. By which what happens, students? The factor number 12, that is your, we can say the Hedgeman factor, at the starting time it is in the inactivate form. When it activates, when it comes to the site of the injury, it becomes activated. Now it is being converted into 12A. You can see, students, I am writing somewhere only the factor number, and some places I am writing number with A. Means what? If I am writing only the Roman letter, you should understand that I am writing only the inactivated factor. But if I am writing A besides that Roman letter, then you should understand that now this factor is being in the activated form. Only factor I am writing, then it is being in the inactivated form. If I am writing A besides the Roman letter, then this factor is in the activated form. Okay, this one inactivate form. Okay, this one inactivate form, but this one is the activated form. Okay, students, no confusion. Only number means inactive form. If A is present, that is the active form. So what happens when the cut occurs? The changes in the endothelial cell takes place at the site of the injury. The factor 12 comes and attach. Factor 12 means the Hedgeman factor comes and attach, by which the activation of the factor number 12 takes place. Now it is being converted into activated Hedgeman factor. Understood? Activated Hedgeman factor, when it will act on the factor number 11, and it will convert the inactivated factor number 11 into activated factor number 11. Carefully listen, students. This factor number 11A, or we can say activated factor, you can say, students, factor 11A, or you can say activated factor. Okay, see, then this activated factor 11 in the presence of calcium, okay, it will convert the inactivated factor number 9, okay, that is your Christmas factor, into activated Christmas factor. Then this the activated Christmas factor in the presence of calcium will combine with the inactivated factor number 8, that is your anti-hemophilic globulin, and it will convert the inactivated factor number 8 into activated factor number 8. Okay, next. This activated factor number 8 will act on the factor number 10, that is, we can say the Stuart power factor, or we can say the inactivated factor number 10, and it will be and this inactivated factor number 10 will be converted into activated factor number 10, or activated Stuart power factor. This activated Stuart power factor in the presence of the calcium will convert the inactivated factor 5 or the inactivated proacceleratin into activated factor number 5. Okay, further this activated factor number 10, activated factor number 5 will combine with the phospholipid in the presence of the calcium, by which finally we will find the thromboplastin. So in this case we are discussing about the blood vessel. In the blood vessel, how the thromboplastin is being formed. Student, why I have written thrombokinase complex? Because in some book it is also written as the thrombokinase complex, but you should not be confused about that. Thrombokinase complex means it is somehow, we can say, it is related to the thromboplastin only. Okay, so students, once more I am telling you, see, cut happens, changes in the endothelial cell takes place. Factor 12, inactivated factor 12 comes and attaches, by which it is being activated into activated factor number 12. This activated factor number 12, okay, act on the inactivated factor 11 to convert it into activated factor number 11. Activated factor number 11 in the presence of calcium convert the inactivated factor number 9 into activated factor number 9. Remember, this activated factor number 9 in the presence of calcium combines with inactivated factor number 8 to convert it into activated factor number 8. This activated factor number 8, okay, it will convert or acts on the factor number or the inactivated factor number 10, and it will convert it into activated factor number 10. This activated factor number 10 in the presence of calcium, it will act on the inactivated factor number 5, and it will be converted into activated factor number 5. Last point, activated factor number 10 and activated factor number 5 will combine with the phospholipid in the presence of calcium to form the thromboplastin, and also it is also said as the thrombokinase complex. Okay students, no confusion. Why it is being said as the intrinsic pathway? Because the formation or the synthesis of the thromboplastin takes place inside the blood vessels. Okay students, please note down. Students, now come to your next pathway, that is your in the tissue level. As I said you, in the blood vessels the pathway is known as your, we can say, the intrinsic pathway. In the tissue level it is known as your, we can say, the extrinsic pathway. So what happens students, let us see. First of all, the tissue will rupture at the cut area, the tissue will rupture, by which it releases the protein having the factor 7, by which the factor 7, or we can say the inactivated factor 7, will be converted into activated factor 7, or we can say activated proconvertin. The proconvertin synthesis takes place basically in the liver in the presence of the vitamin K. Okay, then students, further this activated factor 7 will convert the inactivated factor 10 into activated factor 10. This activated factor 10 in the presence of calcium will convert the factor 5 or the inactivated factor 5 into activated factor 5. Then this activated factor 10 combines with the activated factor 5 to form, in the presence of the which this add means, see, once more I am saying, this activated factor 10, this combines with the activated factor 5 in presence of the lipid which comes out from the tissue. In the presence of calcium, it will form the thrombokinase complex. Okay student, the thromboplastine or the thrombokinase complex. See, further, up to this much we have discussed the thromboplastin. We have discussed that in the tissue level also thromboplastin is being released, in the blood platelet level also thromboplastin is being released. So that is nothing but the thrombokinase complex. So do not confuse thromboplastin, thrombokinase complex, all are same thing. Okay, so this much we have discussed. Rest case is here, we have discussed in the first step. In the first, the general process, what happens? The thromboplastin has been formed, or the thrombokinase complex has been formed. It will help in the synthesis of the, from here students I have written in the shortcut form, because we have discussed in clear in the general process. So in short form I am telling you, the thrombokinase complex or the thromboplastin, after this formation, it helps in the secretion of or the synthesis of the prothrombinase. You know these points. Prothrombinase in the presence of calcium, see, in the presence of calcium, it will convert the prothrombin into thrombin. Okay, and the formation of the prothrombin takes place in the liver in the presence of the vitamin K. Small correction you do in while telling the general process. I have told that from the prothrombinase the prothrombin is being synthesized. Just do a correction. The function of the prothrombinase are of two types. It inactivate heparin, as I have said you. Another the main function is it just converts the prothrombin into thrombin. I have said wrongly that it helps in synthesizing of the prothrombin, but you just corrected that it only helps in the conversion of the prothrombin into thrombin. Okay, students? Okay. But where the prothrombin synthesis takes place? In the liver by the presence of the vitamin K. Okay. Next, you know, it will convert it into thrombin. The thrombin will help to, or we can say by the help of the catalyzation, it catalyzes to formation of the from the fibrinogen, the fibrin will be formed, and finally the fibrin will mix with the blood cells by which blood clot will occur. Okay, students. So in the two way we discussed, in the intrinsic way, in the extrinsic way. Once more I am telling you, tissue rupture occurs, it releases protein by which it combines with the inactivated factor 7 to convert it into activated factor 7, that is the proconvertin. Proconvertin formation takes place in the liver due to the presence of the vitamin K. Activated factor number 7 will convert inactivated factor 10 into activated factor 10. Activated factor 10 in the presence of calcium will convert inactivated factor 5 into activated factor 5. This activated factor 5 combines with activated factor 10, which further combines with the lipid from the tissue in the presence of calcium. Lastly, thrombokinase complex or the thromboplastin is being formed. From the thrombokinase complex, rest is the case we know in the general process. Means from the thrombokinase complex or the thromboplastin, synthesis of the prothrombinase takes place. From the prothrombinase, in the presence of the calcium, it will convert the prothrombin into thrombin, and also it will help in the inactivation of the heparin by which the blood clotting can occur. Next, the thrombin, it will help or catalyzes the fibrinogen to form the fibrin. This fibrin mixed with the blood cell or combines with the blood cells to form the clot. And this prothrombin, where it is being formed? In the liver due to the presence of the vitamin K. Just correct one thing, I have said you prothrombinase, it helps in the synthesis of prothrombin. Just correct this, that the function of the prothrombinase is only to convert the prothrombin into thrombin, and it also help in inactivating the heparin. Where the formation of the prothrombin takes place? In the liver in the presence of the vitamin K. Vitamin K. Okay, students. So, students, today we have discussed completely about the blood clotting and the blood coagulation. Whenever any sort of injury takes place or cut takes place, blood flows regularly, but after some time the blood clot. This process is known as the blood clotting or the blood coagulation. There are 13 factors that are being responsible for or required for the blood clotting. Okay, factor 1, the 2 factor 13, these are written in the Roman letter. Next we discuss the general process of the blood clotting, which consists of 3 important steps. Okay, how from the tissue rupture and the releases the thromboplastin, and the blood platelet also releases the thromboplastin, which is the factor number 3. And finally, how the clot is being formed, we discussed. Next we discussed how from the tissue the thromboplastin or the thrombokinase complex is being formed, and also how from the blood platelet the thromboplastin is also released. That synthesis of the thromboplastin or how the thromboplastin is being formed inside the blood vessel, then it is the case of the intrinsic pathway. If it takes place in the tissue level, then it is the case of the extrinsic pathway. Next we discuss detail in the case of the intrinsic pathway. Next we discuss detail in the case of extrinsic pathway. Okay, so these are the 2 important pathway by which the thromboplastin is being formed. Okay, students, intrinsic pathway and the extrinsic pathway. So that's all for today. We will meet in our next class. Thank you.